Okay, so we've just learnt about protein, the structures that make up quite a lot of our body. Now we'll learn about enzymes, which are a bit more abstract, but it's important you know how they link to protein itself. The first thing we should discuss is the actual function of the enzyme. Enzymes are what are described as catalysts. They speed up a reaction. Imagine how useless it would be if we had to wait an entire day to synthesize a single product, whether that's ATP or whether it's actually digesting food. It would take a lot longer without enzymes and these catalyzed reactions. Now, you might have seen this graph before if you've done GCSE Triple Science, a simple activation energy graph, and you can see as you go up, the energy increases. Now, this is the minimum amount of energy required for an activation to actually start, and that is called the activation energy. What enzymes do in the form of catalysts is they reduce that activation energy, meaning our bodies don't need to be at super duper high heats to go through a simple respiration reaction. The way we initially thought this happened was an enzyme would have a set structure and it would be complementary to a different structure, and those two molecules will come together. The enzyme is one thing and the substrate, which is just any form of molecule that isn't the enzyme that the enzyme can bind to, they would bind together. And at that point, the enzyme would catalyse the reaction of whether it's the breakdown or whether it's the formation of a different product along the reaction chain. The only issue with this model is it suggests that enzymes are perfectly rigid and perfectly structured to the point where the substrate will always fit, no matter what, into this enzyme, which is a decent idea for the most part, apart from the fact that that's never going to happen in real life. You're never going to get a perfectly accurate structure formed every single time to fit every single form of substrate. So what we eventually found out was that these structures, instead of being perfectly tight and rigid, are instead a lot more flexible than we first thought. Our enzymes are instead like a piece of wet clay. They're quite rigid for the most part, but if you shove an object in this case a substrate, it's going to bend around that substrate, and that's what happens with our enzymes. The substrate gets to the point of the enzyme, and the active site, which is where the reaction occurs, the catalyzation of the reaction occurs at least, that begins to change shape to fit the individual substrate. This model is called the induced fit model. The previous one was called the lock and key, and as we've discussed, it is not a very accurate way of understanding the real world. So why is it that one enzyme fits one substrate and these enzymes can change their shape? Well, that is protein. What's actually happening on the molecular level of this system is that the enzymes are changing the tertiary structure, which we learnt about last time, of their active site, which is able to meld around this oncoming substrate beyond the reaches of this specification. The reason this happens is because the forces that are created between the proteins and the substrate that comes in will cause this enzyme to almost consume this substrate and bind to it, much like hemoglobin binds to oxygen to form that protein which can carry oxygen through the bloodstream. Now, whenever you're discussing a question about this, the first thing you should always say about these enzymes is that they're specific. That will almost always get you a mark. The fact that one enzyme goes to one substrate. And as mentioned before, that's because of the way the protein is arranged inside this tertiary structure, inside this active site, to accommodate the specific substrate. Now that we've covered specificity, we'll move on to rate of reactions and limiting factors. So what's a limiting factor? Well, a limiting factor is something that limits the rate of reaction of a certain procedure. Let's look at the metabolic reaction you're probably most familiar with, the basic form of a respiration reaction. So what do you need for respiration? Well, you need sugar, glucose to be precise, And you need oxygen. Now, we've already discussed that this reaction takes place with enzymes, so they are also a consideration for the rate of reaction. And what about temperature? That's something we'll need to consider, because surely the higher temperature you go, the more reaction will occur, because that's just simple kinetic theory, right? Well, we'll come on to that. And the next part you have to consider is, and the next thing you have to consider is the pH of the system, and I'll come to explain why. Some things that are important to realise in this area is the difference between competitive and non-competitive inhibitors, which we'll explain further in a second. So there we've got a couple of things. We've got, first of all, the concentration of the reactants. Now, if we have a low concentration of these, it's sort of obvious that this reaction is not going to occur very fast, because these molecules could be floating around in a vast open space and never touch each other. They'd never touch each other, they're never going to collide at the right intervals to make this reaction occur. So there's one thing, we have to consider the concentration. Now, if we were to reduce the volume of the container that these substrates were in, then you would have a situation where the reaction could be quite fast, even if there weren't many molecules. And as we increase the concentration of these molecules, we will see that the rate of reaction will increase quite sharply to a point. Now, why does it increase to a point? Well, for the most part in biology, the substrate is almost always in excess to the enzyme that is catalyzing the reaction. In a lot of situations, the actual amount of enzyme is quite low, which means most of the active sites are actively being filled by the enzymes. So you don't need that much more substrate for all of the enzyme active sites to be filled, and these reactions to still be taking place while others are trying to get in. Obviously, if something is taking up a space, you cannot occupy that space with something else, even if you've got a million different substrates. If there aren't any active sites open, nothing is going to happen. So that brings us on to the second point, is active site availability, or concentration of enzymes. If we have a lot of enzymes, we're obviously going to have a lot more of the reaction that can take place. And again, this is to a point, but we find that the enzyme point is a lot further along the line than the substrate point, due to the fact that enzymes are almost always less prevalent than the substrates. For this limit to be reached, you would have to have enough enzymes to have their active sites occupied by all of the substrates in that system, which is a situation very unlikely. So what we will see instead is usually the over time graph rather than over concentration graph. And the rate of reaction, while you've got a certain amount of enzymes, will be greatly increased if you add another load of enzymes in, because, as again, the substrates are in excess. When we watch the graph level on either a rate of reaction over time or substance produced over time graph, we will see that the reason this happens is because eventually the enzymes run out of substrates to catalyze the reaction, whether it's hydrolysis or condensation, or, for example, here is respiration in one of the stages, which we'll come to learn about. But when you don't have enough substrate to feed all of the active sites, you're obviously going to have not as many catalysts in your system that are actively working, so the rate of reaction is going to slow. Now, a way we could feasibly speed this reaction up is with heat, right? You might have learned in GCSE chemistry that the rate of reaction is simply the amount of particles that can hit each other in the correct orientation in a certain amount of time. Well, this is true, but when you bring catalysts into the situation, it becomes a bit hazy. As we've said before, catalysts reduce the activation energy, so that's okay for setting off the reaction, but to actually maintain it at a high speed, you're going to need heat in that system. Luckily for us, our bodies are regulated at a 37-38 degree range. This is perfect for these biological catalysts because it allows them to sustain their shape. As we said before, these proteins are still molecules, and when they're in the active sites, if they get too hot, the same thing is if you heated up an ice cube, it's going to melt. In the same way, sort of, with enzymes, if you heat it up too much, it's going to what's called denature, where the bonds that hold them together, which we discussed last time, are going to begin to break apart from each other, and no longer can you have a system where the catalysts are working, because the active site is no longer complementary to the substrate. So it's important that we find that right range in order to maintain these biological catalysts. As I said before, most enzymes in our body work best at the temperatures that our body provides them. In our gut, where it's our core, so it's usually quite warm, these digestive enzymes in our gut will work quite quickly at that temperature and will unlikely to be denatured. Next up is pH. pH is often used to describe a condition that's either acidic or alkaline. If you remember from the previous video, proteins are what we call amphoteric. They undergo reactions in both high pH and low pH. And this also affects the tertiary structure of this system. If you remember, the active site is based off the tertiary structure. If we alter that using a high or low pH, what ends up happening is the structure can become no longer complementary to the substrate that is meant to be complementary to, which stops the reaction from being catalyzed. In much the same way as temperature denatures the enzymes, we also consider this a denaturation of the enzyme. Always refer to the tertiary structure, however. This is the part that becomes complementary. It doesn't matter whether the primary or secondary structure changes as long as the tertiary structure remains the same. But likelihood is, if you do change some of the areas around the active site's primary or secondary structure, it will become a different tertiary structure. Finally, inhibitors. Now, these are a bit difficult to get your head around. I know they are, for me at least. Competitive inhibitors. Competitive inhibitors go for the active site, and the reason they're described as competitive is because substrates are also trying to go there. But obviously, as we discussed earlier, if you've got something occupying a space, you can't have something else be pushed into that space. The same is for inhibitors. Probably the most famous example of an inhibitor is something like cyanide. Cyanide works by binding in the place of a certain chemical, which would usually be assisting in respiration. This certain part of respiration that it would be helping in is called the electron transport chain, and is vital to maintain a constant flow of energy to our bodies. Whereas cyanide just sits here and says, no, you're not working today, and slowly, quickly kills you in a very painful way, and that is because it is a competitive inhibitor. Non-competitive inhibitors, now this is where it gets interesting, not really interesting, just a bit different. They go to a place called the allosteric site, which is on the enzyme. It's a different structure completely, and this is the only time you have to reference it in all of biology, because this site is directly linked to the active site, but isn't the active site. Let me explain. When an inhibitor hits this side of the enzyme, what happens is the active site begins to change structure, almost as if someone was to slowly tighten their grip around your skin and begin to scratch you or hurt you in some way. Your mouth would move, but it wouldn't be because you've directly altered your mouth shape. They haven't gone up to your face and started pulling your jaw down. But nevertheless, you start to change the shape of your mouth, saying, ow, that hurts, stop. It's the same thing for enzymes. If you inhibit this part of the structure, you will have a situation that will alter the shape of the enzyme, and guess what? Will no longer become complementary to a substrate. Examples of these could be certain nerve gas agents which bind to these sites and stop you being able to process the correct minerals from digestion and can slowly kill you, which this time it is slowly because this is usually over the course of minutes, so a long, drawn out, painful death. Not very nice things, but you know, you have to learn about them. The final thing we will discuss this time is reversible versus irreversible inhibition. As we said before, these cyanide things will just sit on your electron transport chain and stop you from respiring correctly. However, there are certain drugs which can reverse this and remove this chemical compound from the enzyme that it is inhibiting. However, unfortunately, there are some irreversible competitive and non-competitive inhibitors. For example, the nerve gas a lot of the time is irreversible, so if you inhale nerve gas and it begins to bind to your enzymes, you will slowly die, and there is nothing people can do about it in some situations, which is a terrible thing and a terrible way to go, but it's mainly because the reaction that takes place is between two reactive and irreversible atoms, such as something like a fluoride or an aluminium, which both are quite reactive, and if they bind in the allosteric site, then we would have to have something more reactive than something like a fluoride or an aluminium, which to bind in that certain area in the correct way, we'd need some form of nanoscience to do, which unfortunately our technology has not caught up to that point yet. But hopefully in the coming years we'll be able to deal with medical situations that result in irreversible damage to people. But until then, we just have to deal with certain things. Next time we'll discuss how to investigate into the rate of reaction of enzymes. And hopefully now you can finally link those 3 factors that I discussed in the first video together.